Thank you everyone for joining today's exciting webinar. The first thing you might notice about this webinar is the number of people on this landing page here. I won't be able to properly introduce anyone in today's webinar, but I do want to say that each of the individuals here are well-respected and established professors and or clinicians in their fields. For those of you that are joining us for the first time and have never joined one of these webinars, I want to welcome you. I'm very excited myself to hear these talks today. In fact, I guess I would say this is probably almost 100 years overdue, ever since the observation in 1925, which I think is the earliest one, at least that I'm aware of, where Warren and Root noticed that the pancreas in type 1 diabetics were smaller. They said smaller than those in non-diabetic individuals. And so that was a really exciting and observation. And ever since then, we've morphed into many different areas. And today we're going to hear several of these. So every presentation today will be about 12 to 15 minutes or so. Before the talk, a few speakers have kindly agreed to set the stage for us and prepare us for the discussion. And then afterwards, we'll have a moderated panel discussion and Q&A. But just briefly, I wanted to share with you that don't know about the Human Eyelid Research Network. This is an initiative that NIH started about 8 years ago. They divided this network up into different themes, and each one of these themes are pictured here as a cartoon working in different areas of human eyelid biology. And we're going to be hearing from groups associated with her, namely from the CBDS, the Consortium on Beta Cell Death and Survival, and the HPAC Consortium, the Human Pancreas Analysis Consortium. But we also have speakers that don't have anything to do with HERN as well. So a really exciting mix of investigators. Really quickly, some of the items that you'll hear today can also be found in the resource catalog. So you can go there and take a look at some of the data sets and or databases that you hear about today. So with that, I want to turn it over to Dr. Raghu Mirmira, who will take us from there.
Okay, I want to welcome everybody to today's webinar. I can see that we've got a lot of attendees, so I'm really excited about that. I want to say that this was, the idea behind this webinar was sort of came about when I had discussions with two of the co-investigators on my CBDS award, Scott Oakes and Manami Hara. And I'm glad that after a few months of planning and preparation, we've been able to put this together. So the way we're going to do this today is I'm going to introduce our moderators for the event, and then they're going to subsequently introduce the speakers, and then you'll hear from everybody towards the end. The idea would be that if you have questions, please put them in the Q&A. And then, of course, towards the moderated panel discussion at the end, if you would like to speak up, that's great. Just raise your hand, use the raise hand function, and one of the moderators will call on you, and they'll open up your mic so you can ask your question live if you prefer. So with that, I want to introduce our two moderators for today's webinar, Teresa Mastracci, who is an assistant professor at Indiana University, Purdue University in Indianapolis, and Scott Oakes, who's a professor of pathology at the University of Chicago. So Scott is an expert in the biology of the exocrine pancreas, and Teresa is an expert in the biology of the endocrine pancreas. So I'm going to turn it over to Teresa. Teresa.
Thank you very much, Raghu. I will share this. So I've been tasked with sort of setting the stage. And I want to thank Raghu and the Hearn for spearheading putting this together, because it's honestly, it's a topic that's near and dear to my heart. It's what we study in the lab. And so I'm very happy that we're starting to see community evolve around the topic of exocrine-endocrine communication and its implications for disease. So in setting the stage, what I wanted to do is remind you of a couple things. One, that as a developmental biologist, we have actually a really nice appreciation for the fact that there is a commonality between the endocrine and the exocrine, and that is that it has a common progenitor cell. The PDX1 positive progenitors that you find in a developing embryonic pancreas will give rise to both the acinar cells as well as the endocrine cells. And so you can see in this little schematic that the progenitor cells will kind of morph into an epithelium, and it's from that epithelium that we have progenitors for both endocrine and exocrine. So if we think about this in a very simple way, we already do have a connection between the endocrine and the exocrine, in that there's that common pancreatic progenitor that leads to a multipotent pancreatic progenitor that then leads to all of the different cell types in the pancreas. And what I'm showing you on the right-hand side is an image we took of an embryonic day 14.5 pancreas. You can see ringed in yellow is the dorsal pancreas, and ringed over here on the right-hand side is the ventral pancreas. And what I wanted to show you with this is that the epithelium makes up a significant portion of the pancreas, and that's where those progenitor cells would come from for both endocrine and exocrine, but there are other cells there too. And so I think one thing we need to start to appreciate, if not in that it's already being appreciated by many people, but how as a community we need to start to understand it, is that the pancreas is a beautiful organ that has many different cell types, and they don't act unique, they don't act as islands. And I think sometimes we maybe think about the pancreas as having distinct functions, or being islands of cells. The islets themselves are considered islands of cells. And though they have these distinct functions of aiding in digestion or aiding in maintaining proper metabolism, they exist in the same structure. And so it's not so outlandish to think that they actually might be talking to each other, and other cells that surround them may also be facilitating this communication. So one evidence of this would be that there is a growing body of evidence that there are many exocrine pancreas diseases that will have an impact on the endocrine and the other way around. And so from this perspective, acute pancreatitis, chronic pancreatitis, cystic fibrosis, EPI, a lot of the individuals that have these more exocrine type disorders end up with diabetes. And then the reverse is also true. As John Cadiz talked about, individuals with diabetes often have, we've observed this reduction in exocrine pancreas size and possibly exocrine pancreas dysfunction. So if you put this all together, we're coming to a place where I think we're appreciating that there's intercommunication between exocrine and endocrine. And so how do we start attacking these open questions in the field? And we have put together a beautiful panel of 4 speakers today that are going to touch on some of these open questions. For example, what's the role of genetics, and what role does it play in the communication between the endocrine and the exocrine? And for that, our first speaker, Gaulton, is going to speak on this particular topic of genetics. We also have a very large open question about anatomical and clinical observations and how that's helping us with an understanding of pancreatic communications. And Martha Campbell-Thompson is going to show us some data that's going to start that conversation. Another open question is, how do the other cells in the pancreas facilitate this communication? And Manami Hara has absolutely beautiful data to talk about how there's integrated pancreatic blood flow. And this is something we really need to start to research more and understand. And then it always helps to learn from other model systems. And so we have Andy Norris, who will be speaking to us on endocrine exocrine pancreatic interactions in cystic fibrosis. And I think that we can learn a lot from that model. So with that, I'm going to turn it over to our first speaker, who is Gaulton. And Kyle is an assistant professor at the University of California at San Diego. And as I just introduced in my previous slide, he's going to speak to us on the genetics of disease in the exocrine and endocrine pancreas. So it's over to you, Kyle.
Thank you so much, Teresa. So I'll talk a bit about our work in understanding the role of exocrine cells in the genetics of type 1 diabetes, and then also to touch a bit more broadly on the role of genetic influences on other forms of diabetes and pancreatic disease. Okay, so I have two broad areas I'll talk about today. Both get at the question of how can we use human genetics to understand this exocrine-endocrine axis in the pancreas. The first is broadly thinking about genetic effects on diseases of the exocrine and endocrine pancreas. And then I'll talk second about variants affecting endocrine cell function that contribute to risk of diabetes. So for the first part, I just have two slides as an overview. So this I put together over the past couple of days, and it gives a kind of landscape across the human genome of loci that contribute to risk of diabetes, either monogenic or polygenic diabetes, or we're calling pancreatic disease, the pancreatitis or pancreatic cancer. And there's numerous loci that affect these diseases, and I put together a list of loci that affect more than one of these diseases. So some of these affect both type 1 and type 2 diabetes. Some affect MODY, monogenic diabetes, and type 2 diabetes, and a handful of other loci affect both diabetes and risk of either pancreatitis or pancreatic cancer. So in total, there were about 30 loci that I found. This is still a running list, so maybe somewhat incomplete, but at least 30 loci that have been implicated in multiple diseases, 7 of which are implicated in both diabetes and a pancreatic disease, and these are highlighted in red: CFTR, CEL, ABO, PDX1, GP2, CTRP1, 2, and HNF1B. And so this gives a clue that there's underlying these loci is likely some shared genetic risk of these diseases of the exocrine and endocrine pancreas. A caveat to this is that it's not necessarily the same causal variant or causal signal at these loci that's contributing to these diseases. In some cases it is, in other cases it may be different variants that are contributing, but perhaps through the same gene. And another caveat is that, of course, for GWAS loci, in most cases we don't at the moment know what the culpable gene is, because most of these loci are non-coding, and it's still quite a challenge to figure out what genes are involved in the disease. But this does give some sense that variants that are affecting diabetes and exocrine pancreas disease in some cases are mapping to the same parts of the genome. Another piece of evidence that supports a general relationship between diabetes and pancreatic disease is that if you look not at specific loci, but you look genome-wide at variants across the entire genome and their effects on diabetes risk and pancreatic disease risk, we see a general correlation in the effects of variants broadly on these two classes of disease. So this was a figure panel from a recent paper in Nature on type 1 diabetes, where we showed that there was a correlation in genetic effects on type 1 diabetes for variants across the genome, of course, on immune-related diseases on the left, but in the middle, we see a positive relationship in genetic effects on type 1 diabetes and type 2 diabetes, as well as pancreatic cancer, and also some evidence for chronic pancreatitis. So that suggests that there's an inherent relationship between genetic effects on these two different types of diseases. So that gives a kind of general overview of both that there are loci, specific loci in the genome that affect diabetes and pancreatic disease, as well as a broader relationship between genetic effects on these two classes of disease. Now, the second part is more about variants that affect exocrine cell function and how these contribute to risk of diabetes, which is some work that we've recently done. And there are three aspects to this section. So to start, I'll touch on things that most are probably familiar with, which are monogenic diseases that appear to affect exocrine cell function that then cause diabetes. One of the best examples is a form of MODY called MODY8, which is caused by mutations in a gene called CEL. It's a rare form of MODY that has pancreatic exocrine insufficiency. CEL is a lipase, a lipid digestive enzyme that's highly expressed in acinar cells. And the proposed mechanism, although I don't know if it's been proven, but the proposed mechanism of how mutations in CEL contribute to development of diabetes is through the formation of protein aggregates that are secreted that then cause beta cell dysfunction. So this gives some indication that genes and proteins in exocrine cells can contribute to the development of diabetes. Another very good example is CFRD, and I think we have talks later on about this disease, cystic fibrosis-related diabetes. Large proportion of individuals with CF develop CFRD. CFTR, which is the gene mutated in cystic fibrosis, is highly expressed in ductal cells. Has been shown to have very limited expression in beta cells, at least in humans. And so therefore the proposed mechanism in nice studies by Al Powers and others argues that the means through which mutations in CFTR contribute to the development of diabetes are through function in ductal cells by increasing eyelid infiltrating immune cells due to pancreatic damage, which then leads to beta cell loss, as opposed to an intrinsic defect within beta cells due to CFTR mutations. So these two examples give a sense for how genes functioning in very specific functions in exocrine cells can affect diabetes risk. So now getting into polygenic diabetes, type 1 and type 2 diabetes. Again, a study that recently came out from our lab was looking at the genetics of type 1 diabetes risk. And in this study, we did two things. So we first performed a large genome-wide association study of case and control samples for type 1 diabetes across the genome to identify variants that affect T1D risk. We identified many loci associated with type 1 diabetes risk. And as like I mentioned before, most of the loci identified through GWAS are non-coding. So if you want to understand their function, you need to map regulatory sequence and overlay that information with these risk variants in the non-coding genome. So to understand how these variants might be functioning, we on the right then performed single-cell attack-seq, which is a genomic technique to map regulatory sequence, and the single-cell version of this technique allows you to derive cell type specific regulatory programs. And we did this in pancreas and whole blood and defined regulatory programs in all of these cell types, which we then combined with the GWAS data for type 1 diabetes. Just one point to note that these resources are available through the HERN resource browser, and then we also have several resources, both for genetics and for genomic data, through which these data can be accessed and queried. So by combining these data together, checking time here, combining these data together, what we realized was that variants that were strongly associated with type 1 diabetes tended to be preferentially located in regulatory elements that were active in certain cell types. So not surprisingly, we found enrichment of T1D risk variants in sites active in T cells, other immune cells, as well as beta cells. But somewhat surprisingly, we also found strong enrichment for risk variants in sites active in acinar and ductal cells. And then when we went and looked at individual loci for type 1 diabetes, there was a subset of loci where likely variants that are responsible for these signals mapped in sites that were active in acinar and or ductal cells, but were largely off in T cells and beta cells and other cell types, suggesting that the variants were mapping in sites that were specific to either acinar or ductal cells, suggesting that the mechanism through which these variants contribute to type 1 diabetes risk is by affecting exocrine cell regulation. If we then look at what genes these loci are nearby, many of these loci are either linked to or very close to genes that have highly specific expression in either acinar or ductal cells. And so here are some examples of these loci where there are risk variants for type 1 diabetes, again, non-coding, that are in regulatory elements active in acinar ductal cells that are then linked to genes with very specific expression in those cell types. One example is CTRV1 and 2, which are digestive enzymes. Another example is GP2, which is a component of the zymogen granules produced by the acinar cells. Another example is CEL, which is the same gene that causes MODY8, which has the purported exocrine insufficiency and exocrine mechanism. And so all of these loci are linked to genes that have a very specialized function in acinar cells. Another locus that we found, which was quite interesting to us, which was exocrine specific, were variants that mapped at the CFTR locus. So the risk variants for type 1 diabetes map upstream of CFTR. in regulatory elements that are active only in ductal cells, not in other cell types. And these variants are linked to CFTR in ductal cells, and we confirmed that this element regulates CFTR expression in ductal cell models, and also the same variants that affect type 1 diabetes risk are also correlated with the expression of CFTR, specifically in ductal cells, and the risk variant, or sorry, the risk allele for type 1 diabetes is correlated with decreased expression of CFTR, suggesting that decreased CFTR in ductal cells increases risk of type 1 diabetes. So then that led us the question of how do exocrine cells contribute to risk of type 1 diabetes. As John and Teresa have said, studies have long reported abnormalities of the exocrine pancreas in T1D, but the question of how do genes that function in exocrine cells contribute causally to the development of T1D is still open. Many exocrine genes implicated in T1D are digestive enzymes or involved in their secretion, the genes that I mentioned. And also T1D variants at these loci are generally enriched for association with pancreatitis, other pancreatic disease. CFTR as an example, so this variant at CFTR associated with type 1 diabetes, also associated with pancreatitis and other pancreatic diseases. So we hypothesized that the mechanism through which these variants contribute to T1D in the exocrine pancreas is by promoting inflammation, immune infiltration via subclinical inflammation. In the case of CFTR, that would be similar to the proposed mechanism for how it's contributing to the development of CFRD. Okay, so very quickly, there are also some examples for type 2 diabetes that I was tangentially involved in, and others and people I've worked with previously, where risk loci associated with type 2 diabetes have also been suggested to affect exocrine cell function. A very good example of this, again, is CTRB1/2, which is also a type 1 diabetes risk locus, also affects pancreatitis and I believe pancreatic cancer. In this case, the same variant that's causal for type 1 diabetes is also causal for type 2 diabetes. And what's interesting about this locus is that they have the opposite direction of effect on these diseases. So the allele that increases risk of type 1 diabetes is protective for type 2 diabetes. So it is an interesting mechanism underlying this. Risk variants for type 2 diabetes associated with levels at CTRB1, as well as in trans levels of other digestive enzymes in acinar cells. So how might variants affecting exocrine cells contribute to type 2 diabetes risk? For type 1 diabetes, we propose through subclinical inflammation and immune infiltration. For type 2 diabetes, perhaps it's more through signaling to the gut, which then signals back to beta cells. Some evidence supporting this is that for GWASs for 2-hour glucose in response to OGTT, we see strong enrichment of variants associated with 2-hour glucose in acinar and ductal sites. And also these variants are strongly enriched for genes that are expressed in both the pancreas and the stomach. So we hypothesize that perhaps the effect on type 2 diabetes is through the release of these enzymes into the gut and then signaling back to beta cells via the incretin pathway. And finally, there's also some evidence that of direct effects on beta cells by exocrine function for type 2 diabetes. A really cool study found rare coding variants in this gene CELA2A, which was associated with metabolic syndrome, type 2 diabetes, cardiovascular disease. CELA2A is a circulating enzyme produced in acinar cells, and this study showed that this protein affects the regulation of insulin secretion and insulin degradation, likely through direct effects on beta cells. Okay, and a lot of this data can be found in the genetic space on these resources, which are also listed in the HERN resource portal. But not to belabor that. So to summarize, 2 points. So genetic effects on exocrine endocrine disease through shared loci that affect both types of disease, and also a broad correlation in the effects genome-wide on these classes of disease. And 2, variants affect exocrine cell function, contribute to diabetes risk in both T1D and T2D. In T1D, potentially by promoting inflammation and infiltration of immune cells. T2D, potentially by affecting beta cell function. And in secretion, both indirectly via incretin in the gut and also directly on beta cells. So thank you.
Fantastic. That was a great talk, Kyle. Thank you so much. So if anyone has any questions for Kyle, you can put those into the Q&A now. If you'd rather ask Kyle the question directly, you can raise your hand at the end of all of the speakers and when we have our moderated panel discussion. So I'd like to invite Martha Campbell-Thompson to share her screen. She'll be our next presenter. She is a professor at the University of Florida, and she's going to speak to us on the clinical and histopathological correlates of endocrine-exocrine communication.
Great, thank you. And Layla, it says that you need to show me as the host. That was a beautiful introduction by Kyle. And so we're going to go now kind of from the bench to the bedside, I hope, and show you some correlates of what we see histopathologically as evidence of this communication. No disclosures. I study primarily type 1 diabetes, and type 1 diabetes is being increasingly appreciated as being more of a spectrum of diseases. While originally called juvenile diabetes, we now believe that over half of new patients are actually adults. But I've always appreciated the idea that development of the disease could start in utero, or even epigenomic effects on one's father or mother. And through a combination of hits, whether they be through your genes or exposure to certain microbes, including viruses, your beta cells may be attacked. And whether or not you go on to develop diabetes depends upon your inherent resistance to this. A reminder, as if we need one though, that the endocrine compartment of the pancreas represents only 1 to 2% of its volume. A small pancreas was actually first described in 1909, where 25% of the type 1 subjects had smaller pancreases. And then through the years, many people have described this phenomenon. The pancreas grows through early adulthood up until about 20 years of age, and then plateaus. And then after age 60, it starts to decline. And this may underlie some of the association of increased risk of diabetes or even pancreatitis with aging. And normal aging phenomenon have been reported as acinar atrophy, fibrosis, and fat infiltration. We initially reported in 2012, along with John Cadiz, that the pancreas was small in subjects, organ donors in the NPOD program. And this decrease was about 25% compared to the normal pancreas weight. And we showed it, as others have, that there was this decrease in type 1 organ donors compared to controls. As I showed previously, our pancreas grows. Certainly, we also grow. This work was published in 2016, and the top panel shows as one's age increases, the body weight increases proportionally. So they're very closely correlated in people without diabetes. The middle panel shows that from the autoantibody positive organ donors at the time. And then the bottom panel shows how type 1 diabetic organ donors have a normal body size, but their pancreas weight is half that of normals. So from this, we started looking at the ratio of the pancreas weight to the body weight. And looking at the pancreas, not from the whole organ, but rather divided into the three main regions, head, body, and tail, we saw this decrease in size from head, body, and tail in the type 1 diabetic donors and also the type 2. Conflicting reports show that type 2 diabetes rather is associated with a small pancreas size, but these are confounded by the usual presence of fat in the pancreas. So it's a little bit more difficult to tell whether the size is decreasing in type 2. Nonetheless, they also show this decrease between head, body, and tail regions. And this is shown in the next slide, images taken during processing of the pancreas by the NPOD lab. And in the top panel is a controlled pancreas shown from a 14-year-old male compared to that of a 13-year-old male. So you can see this decrease in pancreas size. And it's related to the ventral and dorsal lobe regions of the pancreas, as Teresa mentioned, that the ventral lobe of the pancreas is smaller, and during rotations along the body axis during growth of the fetus becomes fused to the head region. So what we are observing is a loss in the dorsal lobe region. How does this change, or does it change over duration of diabetes? Again, from the NPOD program, a cross-sectional study, we do see a slight decrease over diabetes duration. But I'd like to emphasize at the 0 point, at onset of disease, the relative pancreas weight is already decreased. And so it's a small pancreas at onset of disease is observed. This prompted us to do a pilot study through the TrialNet program, using MRI, and all of the preliminary studies were conducted at the University of Florida on one scanner to eliminate potential confounding variables associated with multi-sites. And again, the image shown on the lower left shows a control subject versus a type 1. And then on the right side, we see a 3D amalgamation of the 2D slices. And we're using deep learning to reconstruct the pancreas in 3D, not only to obtain this overall volume, but to go deeper and to see, can we actually, through deep learning methods, reconstruct some of the histology that's going on in this disease. In 2019, we published the results of that study, and it showed that if you are a first-degree relative of someone with type 1 diabetes and you're autoantibody negative, you actually have a small pancreas, smaller compared to autoantibody negative control subjects. And furthermore, if you have single or multiple autoantibodies, you have an intermediate pancreas size between the autoantibody negative first degree relatives and those with type 1 diabetes. We had fairly small sample sizes, but even with that, we could show that as you go from negativity to single, multiple, 2, 3, 4 autoantibodies, you're trending downward in having a small pancreas size. Our subjects were examined one time during the first year of onset, and during this time we didn't see a change in pancreas size, nor did we see a change in the exocrine pancreatic enzyme trypsinogen measured in the blood. But work at the University of Diabetes Institute is ongoing, and this work was published in 2017 showing two things. One, that we could replicate the decrease in serum trypsinogen in recent onset T1D, and also, interestingly, an elevation of trypsinogen in the type 2 subjects that were studied. But importantly, age was looked at. And here on the right-hand side, you can see this increase in serum levels of controls. So the serum trypsinogen is mirroring the pancreas size. And again, this loss of this in T1D subjects. This has been replicated by others working in the field. In Australia, the India study. And here, instead of looking at the serum, they looked at fecal elastase. And they had at-risk children and two groups, progressors and non-progressors. And what is shown that in those at-risk children, if they did not progress, they did not see any change or relative change in fecal elastase. But indeed, in those that did progress, shown in the lower right panel, their fecal elastase decreased. So that we now have another study ongoing with first-degree relatives through the TrialNet program, where we're going to be looking at longitudinal pancreas size at 0 and 12 months, and with multiple correlations to certainly beta cell function, but also endocrine cell function, and also genetics of the host. We're again using MRI, but this is a multi-center study. Now it will be done on 3T magnets, and the first group to start will all be using Siemens Prisma or Skyra scanners. And here are the current sites getting ready to go, including Australia. So the relative change is relatively stable in controls, as we showed, up to 7 years. And also in our multiple autoantibodies, we show some increase, but the majority of them decreased. Longitudinal imaging in type 1 subjects and autoantibody subjects is also of great interest to Jack Vorosky and Al Powers at Vanderbilt, and they published in 2019 this decrease in type 1 size over a 12-month period. We've also done some longitudinal imaging in T1D subjects. Here, note the differences in the x-axis, where we are plotting it by months post-onset, whereas theirs is showing it from the first measurement. That's the clinical evidence I'd like to show. And now we move into the histopathology of type 1 diabetes. Again, from the Vanderbilt group shown in 2020, that the size of acinar cells is not different. And here they looked at cells close to the islet versus in the periphery, and this has been shown by other groups, and also between the three regions, that the acinar cells in the tail are not smaller. But what they did show was that the numbers of cells, if you take a cross-sectional section from the pancreas, you have fewer cells in type 1. So it is a decrease in numbers of acinar cells producing the enzymes that we may be seeing peripherally. They also showed, using trichrome staining, that there was an increase in that in the type 1 subjects. And then this was also shown by a group in 2018 using image analysis from the online pathology system of NPOD, what they called non-exocrine. So this would include ducts and also extracellular matrix. And they showed in this patients or donors with T1D that it was an increased proportion of this exocrine area, or non-exocrine they called it. And again, looking at age effects, so that the controls increase, but so do the type 1s, but they're lower. The group here at the University of Florida has also looked at the duct system and numbers of acinar cells and the like. And here shown on the left in work that was published this year, they looked at the area around the duct and as expressed as a proportion of the cross-sectional area. And they looked at also autoantibody cases and divided the T1D group into those with residual beta cells or without. And the acinar area was only decreased in the insulin negative type 1 subjects, shown in C. And furthermore, again, this was most prominent in the tail region. My lab has looked at the eyelid inflammation, and here we see a correlation, a positive correlation between exocrine and endocrine inflammation, including that if you have higher numbers of insulitic islets, you have more inflammation in the exocrine region. And Matthias von Harris group and Teresa Rodriguez Calvo have also looked extensively at the CD8 cells in T1D, showing that there is a higher number of CD8 cells in the exocrine pancreas. And in panel A, in the middle portion, again, we see the tail region of interest. And then others have also shown an increase in neutrophils. So we have a pro-inflammatory environment. My group has also looked at the islet microvasculature in type 1 diabetics compared to controls. And the vessels within the islets approximate that of the exocrine region, where we don't see a difference in terms of diameters or density between controls and type 1. And moreover, again, if you have T1D subjects without insulin, without beta cells, they have a smaller diameter. This also correlated actually with the diabetes duration. And again, this has been published. So in conclusion, the relative pancreas size and potential, the longitudinal change in size, may serve as a new biomarker for determining someone's risk, so-called personalized risk score for someone. And it also may help us understand the disease heterogeneity, or so-called endotypes. The dorsal lobe region, particularly the tail region, shows the greatest changes in this. And we certainly think that abnormalities in the exocrine microenvironment may underlie beta cell heterogeneity that's reported, and certainly impact their survival and regeneration. So thank you very much, and I turn this over to Teresa again.
Yeah, thank you, Martha. I'm going to pick up from here. That was a beautiful talk, and I'm sure there's going to be lots of questions in the question and answer session afterward. Our next speaker is going to be Manami Hara, who's a research associate professor from the University of Chicago, and she's going to talk about blood flow between the endocrine and the exocrine pancreas. Manami, you able to bring up your slides? Wonderful, and then just put them on presenter view. Excellent. You're still on. I think I still see your. Yeah, there you go. Perfect. Take it away. Manami.
Okay.
There you go. Okay.
Okay. Okay, thank you. So islets have been considered isolated microorgans independent from the exocrine pancreas. And when it comes to the islet blood flow, I believe these illustrations represent the general view that blood flows through our feeding arteriole and drains out from one or more venules. So it's one-way traffic. So if that's so, have you ever thought about why experiment using isolated islet work? So shown here is a human islet cultured for 4 days. And sustained with insulin, glucagon, somatostatin, and CD31 on an endothelial cell marker. So as you see, all the capillary and entries, as well as peripheral islet cells exposed to whatever you give them. Then what about live pancreas tissue slices that have been recently introduced to the field? So if you believe this is truly the pattern of islet blood flow in vivo, how would anyone be able to record such beautiful responses to glucose, for example? So to say the least, it would be very difficult to reach the feeding arteriole within a thick tissue slice in vitro. So here today, I'd like to propose a new model of the pancreatic blood flow that explains why these two experimental systems are relevant, and they actually mimic the in vivo situation or settings. So first, through collaboration with these investigators at the University of Chicago, we analyzed intravital recordings of almost 400 islets from 200 mice. And here is large islet from a MIP-GFP mouse. So these are di-eye labeled red blood cells or RBCs. So here we are only showing RBCs. And then using image analysis software, IMARIS, individual RBCs are digitally captured. Then you can do all kinds of analysis like tracking each individual RBC movement. And you just let them go throughout each recording so that the tracks will be accumulated. Then you see all traces with heat map. So here, red to white depicts slow to fast. And as shown here is multiple directions of blood flows by arrows. Then we quantify the direction of blood flow at the interface. So blood flow direction from endocrine to exocrine is marked by pink arrows, and from exocrine to endocrine is marked by blue arrows. So the ratio of the two directions was about 1:1. So now this is a human islet in situ, stained with the pan-endocrine cell marker in cyan. The same image here is only showing vasculature. So as you see, the integration of so-called islet capillaries is around here, and surrounding tissues is at its entirety. So here, the islet is made transparent, so you can see the connection. I mean, they're all continuous. And next, in this fluorescent image, alpha-smooth muscle actin or SMA labeled arterioles are shown in magenta. And they are all 3D surface rendered, and arterioles are in blue. So looking over many 3D images like this one, I sort of reached the point that I couldn't really ignore the existence of islets that have or like this one, no contact with arterioles. So we examined the spatial relationship between arterioles and islets in thick pancreatic tissue from various species. So this is a human pancreas. These islets are labeled from A to H that are corresponding here. So these islets are captured from different angles. So they don't quite look like what we knew as feeding arterioles, do they? So maybe something like B, but others are really quite random. So similarly in monkeys, you can see there's no really feeding arteriole. And then now this is pig, rabbit, ferret, and a mouse. So here we propose the pancreatic arteriole feeds the whole pancreas regionally, not targeting individual islets. So let me explain. So here's the fluorescent image that's you know, kind of large scale, and then these are fluorescent, so they are rendered so that you can see the structure really well. So these large vessels branch into arterioles. And then overall islets are in a close proximity, but not necessarily, you know, all under the arteriole, like a 1-to-1 ratio. This may be something like a feeding arteriole, but nothing here. And these are islets, you know, basically just catching, okay, except this. And this is relatively rare, I would say. And then so they can just, you know, really kind of bi-arterial. And really surprising thing, I mean, to me, I'm going to show it again, but these capillaries can branch out directly from the arteriole. And then so they kind of reside around arteriole, but not necessarily, you know, receiving so-called like a dedicated feeding arteriole. So something like this. And then again, really surprised, I mean, unexpected thing is that the capillaries can branch out directly from the arteriole, so that, so I kind of always thought a capillary only come out at the terminal point of an arteriole, you know, kind of suggested here, but actually these arteriole are just really passing through. So if islets are just touching like this, then they can see changes of blood glucose, I mean blood flow from capillaries at first hand. So overall, the arteriole emerged to feed the exocrine pancreas regionally. And vascularizing the pancreas in this way may allow an entire downstream region of islets and acinar cells to be simultaneously exposed to changes in the blood levels of glucose, hormones, and any other circulating factors. Then, of course, I would say it's not fair not referring to this seminal article by Drs. Bonnewier and Oates, published in 1982. So this is the figure 1 of the article. And in this beautiful collagen cast image, they labeled this as a large arteriole. And then this one and this one, like here, as small arteriole. But in fact, they are capillaries. And then I would think this interpretation probably came from this group's work in 1970s, who pioneered vascular collagen casting in various tissues. So they first showed the rat renal glomerulus. So it's afferent arteriole and efferent arteriole. So they are large, you know, way larger than capillaries. Then in 1973, same group showed monkey pancreas, and then they called this little thing so vas afferens. So that's our arteriole. So here, to be fair, I'd like to point out that recent advancement of technologies, including microscopy, imaging software, tissue clearing, and a specific cell markers such as SMA, just made all the current findings possible. But nonetheless, I'm glad to know that after all, we are looking at the same thing. So finally, since we found that many islets do not have a contact with arterioles, we wanted to see if there's any differences between islets with and without contact to the arteriole. So here we, so here arterioles are shown in blue, and these are islets. This is the imaris. So they can actually identify each with ID numbers, and it comes with all other measurement as well. So we measured shortest distance between islets and arterioles. So simply that if there's a contact, it's 0, and no contact, than positive values. So here, eyelid contact in the arteriole are significantly actually larger in size than those had no contact. So they're color coded here, and a contact in one is in cyan, and those with no contact is in red. So isn't the difference in eyelid size obvious just by eye? And then it was actually true to all species that we studied as well. The eyelid volume is converted to effective diameter. And these are contacting eyelids and the no contacting. So you can just compare mean contacting one significantly larger than those that had no contact. And also eyelid numbers are larger, way larger than those contacting ones. So in conclusion, our findings suggest a new model for pancreatic microcirculation that allows a much more intimate communication in the blood flow between the exocrine and the endocrine pancreas than currently recognized. And I think the fact that isolated islets and the live pancreas tissue /experiment work is the evidence that support this model of pancreatic regional blood flow. Thank you for your attention.
Thank you, Manami, very much for such a beautiful talk and images. Unfortunately, Andy Norris is not going to be able to be our fourth speaker for reasons beyond his control. He won't be able to join us today. And so, but that's okay. I think it will give us some more time for discussion and the question and answer session. So I'm going to hand this back to Teresa, and the two of us together will try to field questions.
That's great. Thanks, Scott. And those were wonderful talks. So I guess at this point, we're opening it up for questions. If anybody has a question, you can put it into the Q&A. We do have some already in there. And if you'd rather ask your question live, I believe you can use the raised hand feature, and we'll get to you when we see you. So if we start with Kyle, there's a question from Rohit Kulkarni in the chat that says that he's curious about the secreted factors acting directly on the beta cells. Does elastase directly impact secretory pathways, and if yes, which elastase?
So in the paper that found mutations in cell A2A, they looked at effects on introducing cell A2A protein in culture, I think beta cells, and they showed, I believe, activation of protein kinase A. So that may suggest like some AMP related effects, and then also showed increase in expression of calcium channels, and then increased levels of calcium in beta cells. So I believe that was the extent to which they looked at intracellular effects in beta cells in that study. Beyond that, I'm not sure, but that's at least what they found in studying that gene in that study. I don't know if Teresa, you know more than that, but
No, actually, and I have had a similar question to Rohit's actually, but kind of taking off on that, the idea that we are, and this kind of goes with some of what Martha showed us too, that there's this increase potentially or decrease in the case of type 1 diabetes of enzymes. And so if we go back to the idea of the communication, Kyle, you're linking this, you think, to an effect on inflammation. Do you think that, you know, that's the only potential effect that alterations in the secreted enzymes could actually have on the beta cells or other cells, the duct cells, for example?
Right. Probably not. I think that what we've seen and our thoughts in terms of thinking that these genes may be contributing to type 1 diabetes risk is largely through they're also being associated with pancreatitis risk and in some cases pancreatic cancer. Which I think seems plausible that is one way that they're contributing to type 1 diabetes. But I think beyond that, it's entirely possible that they're signaling directly to beta cells. That's not something we've looked at, nor I think had the ability, at least in terms of the genetic data we had, to ask in that study. But I think that seems entirely plausible. I don't know if likely, but that seems like it's probably not the only mechanism through inflammation. Yeah.
Martha, is that something you think you can interrogate in your samples and your system?
Possibly, but I think it would be better done through a combination of patient samples and in vitro models, or in vivo living pancreas slices in a way. So
yeah, I think your work has addressed the idea that obviously the number of cells has changed, and so we can have the expectation that what's made by those cells would decrease. But I guess there's still the outstanding question of, are those cells actually secreting properly?
Exactly.
The ones that are left.
Exactly.
Yeah.
Their function.
Yeah. So there's actually a question for you, Martha, in the Q&A from Giorgio Basil. I apologize if I said the name incorrectly. Are there autoantibodies against exocrine-specific proteins in type 1 diabetes patients? And do you think that the infiltration of the exocrine compartment from immune cells is a consequence of eyelid infiltration, or is it specific?
Yes. Great question. I'd like to think that it could be both, that there may be an attack on the exocrine pancreas. So many people have looked at the demise of beta cells from a beta cell centric view of type 1 diabetes, and I certainly most of the time still do. But what if it starts in the exocrine pancreas? What if, as the null hypothesis to the beta-centric model? Certainly there's inflammation in the exocrine pancreas. I think it's primarily the Japanese studies have shown some exocrine autoantibodies like carbonic anhydrase 9, which is a kind of mature duct marker, and lactoferrin. There's even the work of others that have shown autoantibodies to peripherin, which is a neural marker. And I would love for the immunologists to look at autoantibodies more in type 1 diabetes. And there are groups working on this. But again, as with type 1 diabetes, are they a consequence of exposure of cell antigens rather than having a pathogenic role of any sort?
So I wanted to jump to Manami because I have a question for you. I think your work is beautiful. We do have more questions for both Kyle and Martha. We're going to come back to them. But I think your work is beautiful in that it definitely underscores how when we have newer tools, sometimes this can help us understand what we previously thought about from static images. And, we can kind of bring the ideas that we understood from the static images to life in these, using the new tools. So you said that the islets that do contact are larger than the islets that don't contact. And you think that the lack of contact is the reason for the size change, the size difference? Knowing what could be in the blood, including insulin, right? That the islets that may have direct contact with the bloodstream and the flow might actually be larger in size and contain more cells, and could the lack of contact?
Yeah, I think it takes on more kind of developmental biology from fetus to neonate, maybe not so much adult, I guess, and then that's exactly what we started doing, right?
Yeah.
So actually, Teresa's, I saw the question from Dorian as well. So they've been sending me fetal pancreas tissues, and then as he asked, we are also, Teresa too, interested in vasculature and duct And then as you suggested, they are really close by, and they are actually really, really close. But I don't think anyone really looked at it in a large scale. So in the movies I showed, the images I showed, those humans those are totally adult. And then I think that's kind of end stage. You just have that resulted in like that. So, probably they, in the embryonic, in the embryos, all the acinar tissues and those vasculature kind of grow together. So they should be way more closer. Everybody should be close to their vasculature, and they just expand pancreas, and then the summer just kind of stay comfortable without touching arterioles anymore. And then I think the blood flow is really rapid, like regionally, not whole the pancreas, but it just happen boom, boom, boom. So I don't think in the end it doesn't really matter how close you are. Maybe you respond like electrically respond faster, but that's kind of milliseconds, seconds order. So I don't really have the answer yet.
Oh, yeah.
These are all guessing.
Exactly. I think what a lot of your videos do is help us to pose new questions now that we can see sort of the islets in a different, dimension, I guess.
Yeah.
So Rohit, you have another question here for Martha. If you want to raise your hand, then we can find you and unmute you. There we go. So you can unmute yourself, or you'll be unmuted, I guess. And please feel free to ask your question.
Yeah, great. Thanks. First of all, a great panel and excellent talks. And obviously, we missed Norris's talk. But so, Martha, very exciting stuff. But I'm just wanted to get your opinion on what it means. So, having a reduced volume in the T1D patients of the pancreas. So what? And so the question is two parts. One is, are there other organs in the T1D patients who also show a reduced size? And if whatever that outcome is, but have you guys also looked at, do these patients show reduced growth hormones? Is there generalized alterations in the organs? Because I just don't know. I know that this keeps coming up all the time, that T1D has a reduced volume. And the question is, so what?
Yes, well, exactly. So what? So for me, I think it, again, could inform one's risk of developing type 1 diabetes. And so if you have multiple autoantibodies, you're a ticking time bomb in terms of when are you going to develop type 1 diabetes? Through imaging, that could be assessed, and it may inform us about someone's risk. And if you have multiple autoantibodies and you have a normal sized pancreas, there may be different therapies or even anti-inflammatories or whatever. The treatment could be different. This could help also explain some of the variability that clinical trials have for in TrialNet. And so I think it's another parameter to evaluate in someone with type 1 diabetes. Are there other organs? As I mentioned, someone's body size is normal. We haven't absolutely imaged or measured other organs. There are other organs in the plane of these MRI that could be measured. There's some reports that with diabetes, both type 1 and type 2, that the thickness of your cortex is smaller. So insulin is a growth factor for the brain, for many organs. And in terms of IGF, Melanie Shapiro here in Todd Brusco's lab has looked at the IGF levels in our DP3 subjects and did not see a difference. But those studies are certainly ongoing. Insulin and IGF share a receptor, and it's a very interesting question that needs to be looked at in more depth.
Because I'm just wondering, just to last comment, that whether one can experimentally induce diabetes, because we still don't know what is the cause, right? Cause or effect of this smaller volume. So can we design an experiment where in a mouse model, transplant a smaller volume of the pancreas versus a larger volume of the pancreas and see whether that leads to, some defect in glycemic control? I'm just thinking out loud here to see what it's.
Well, those transplants, it might be interesting to vary numbers of islets and acinar cells that are transplanted. So if you transplant more islets with the same acinar number of cells, how do they possibly change? That might be a corollary to that experiment.
Thank you.
Thank you, Rohit.
So I'm going to jump in here.
Yeah.
There's a bunch of good questions still. I was, some of these cross a few themes that I love in particular. There's Katie Coates' question. I don't know, Katie, if you want to ask it yourself, or I'll read it if you don't, but if you want to unmute yourself.
Sure, I'd be glad to. Thank you. Can you hear me?
Yes.
Excellent. Yeah, something kind of struck my interest several months ago. There was a paper published in Cell Metabolism back in February. It was a liver paper, but they showed that through connexin 34, there was intracellular transmission of ER stress in hepatocytes. And we know from the importance of connexin 36 and other channels, and from Richard Benninger's work and many others, about the role of that in kind of propagating calcium signals across the islet. And I was thinking also about how ER stress is common not only in the endocrine pancreas, but also in the exocrine pancreas and many different diseases. Is there a possibility that there could be intracellular transmission of ER stress in these different cell types within the pancreas that could be impacting each other's function?
Yeah, it's a question very, very near and dear to my heart. Raghu, you've I'm sure thought of this, and this Hearn grant is in some ways dealing with that. Do you want to weigh in?
Yeah, well, I think that's a great question, Katie, and I think the million-dollar question really is, how does ER stress work in the context of crosstalk? I think there's a lot already there, and definitely, Scott, you could speak to the role of ER stress in the exocrine compartment. And I think we've talked, particularly within the Hearn, a lot about ER stress in the endocrine compartment. The question now really is, how does stress, intracellular stress pathways like ER stress in one compartment communicate to the other? And I think this is really a million-dollar question. Are there other signals that can be transmitted from one compartment to the other? We've already seen where signals can get transmitted within the islet. For example, the senescence and the SASP response within the islet. Some nice work by Anil Bhushan's group, but it does beg the question, and I don't know if you had any thoughts about that, Scott.
Yeah, I mean, just to expand the possibilities here. So from different